Hello, everyone. Thank you for joining us today for this second webinar. Today, Adam Mesian will be presenting drug encapsulation in biocompatible microparticles for drug delivery. Just a few notes before we get started. If you have any questions during the presentation, please use the chat panel available from the top right side of the screen. Our team will answer them right away if possible, or we'll bring them up during the presentation during the question station. A recorded version of the webinar will be available from tomorrow. The presentation is also available from you to download along with other documents. Due to the current situation, this webinar is recorded from home. We hope not to encounter any technical difficulties. However, we apologize in advance if it will be the case. I think that we have most people here, so we get started in 2 minutes. And Adam, I'll give you the lead.
Okay, thank you, Bastien. Hello, everyone. I'm really happy to welcome you on this webinar. So today we're going to see how we can use microphysics and most exactly droplet-based microphysics to encapsulate drug inside biocompatible microparticles. So what you will learn during this webinar, you will learn a new method for microparticle production. What is droplet-based microphysics exactly? For those who are not aware about microphysics, how we can use droplet-based microphysics to encapsulate drug inside microparticles, and you will understand the benefits and limitations of this technology. So first, let me introduce you the team who is leading this webinar today. So first of all, myself, Adam Mesian. I'm working as project manager at Fluigent, and my work is focused on droplet-based microphysics, how we can use this method to many applications and mostly in pharma application. Then we have Adrien de Wand. Adrien de Wand is a technology lead at Sequoia Technologies. Sequoia is a spin-off from ULB, Université Libre de Bruxelles in Belgium. And Sequoia is using microphysics to develop new technologies for precise production of pharmaceutical projects. We are working with Sequoia in collaboration to overcome all the challenges in pharma. And then we have Arno Reitz. Arno Reitz is a project manager at Fluigent. Arno Reitz has a strong expertise in droplet-based microphysics for every kind of application. So first of all, let me give you a little bit of context regarding the encapsulation in pharmacology. So current treatment in pharma actually show many limitations, such as untargeted delivery. A good example is in chemotherapy, when the treatment is affecting tumoral cell, but it is also affecting other organs and the whole body in general, and the patient could suffer from side effects. Another limitation and big limitation is the poor control of the drug release and the short-time release. Indeed, when we are taking drugs, actually a big amount of active ingredient is released into the body. We are talking about burst release effect. And this release is made for a really short time. So to make sure that every patient receives a good amount of drugs during the whole day and every day, the only solution that we have today is to increase the number of administration. By increasing this number of administration, we're increasing the burst release effect, and then this could lead to side effect and to psychological effect as well. But what if we could develop drugs that could target specific sites, specific organs, control really precisely the delivery of the drugs during a specific amount of time, deliver the right amount, not less, not more, and for a long period of time to avoid dose frequency. Encapsulation of drug in size-controlled particles answers this question and is a way to solve all these issues that I presented. By controlling encapsulation into microparticle really controlled in size, we are able to provide a long-term delivery, deliver the accurate amount of drug that is needed for patients, which leads to a reduction of dose frequency. So when we are talking about encapsulation, there is 3 different levels. So nano encapsulation for nanometric scale, encapsulation into nanoparticles, microencapsulation for encapsulation into microparticles, and capsule for encapsulation at millimetric scales. Today, I would like to focus on the micro-encapsulation and mostly encapsulation into microparticles. So why are we using these microparticles? Microparticles are widely used as drug delivery system. Why? To protect the drug into biocompatible material from the environment, oxidation, temperature, difference of pH. This allows to target specific sites and avoid degradation between the transport, physiological transport. Then it allows to provide accurate delivery of small quantities of drugs by controlling the particle size and the concentration of the material used. We can control the delivery of the drug by small quantities, control the release over a long period of time from one single injection, which can lead to a reduction of dose frequency. But how do we make this microparticle? How do we encapsulate drugs inside microparticle? There is many methods to produce microparticle, such as spray drying, jet milling, but I would like today to focus on the emulsion in batch because this is the most widespread method. Traditional batch method consists of use two immersible fluids into a reservoir to generate forces. It could be sonication, it could be mechanical forces, and this will lead to a generation of emulsion of microparticle generation into another liquid. So there is many advantages to this technology, such as a large-scale production. We are able to generate really large amount of microparticle microemulsion from kilogram to ton per day, for example. This is a conventional method which is widely used and widespread in industrial pharmaceutical area. And also it is FDA approved. So according to Food and Drug Administration, it is approved. This technology is approved. However, we have seen that this technology appears with many limitations, such as a big polydispersity. That means that we have a big dispersion in terms of microparticle droplet size. And in this application, in drug delivery, we want to target one specific size because the size of the microparticle is directly related to the delivery of the drug, the release of the drug. So to target a specific size, we need to add a post-treatment method, such as sorting, and only keep a range of droplets and get rid of the other. So we will also generate a loss of expensive reagents. There is no control on microparticle size, as we have a big dispersion in terms of size. Then poor batch-to-batch repeatability, as we do not control the microparticle size. By using this method, we have also low encapsulation efficiency. And by using mechanical forces or sonication, we are generating uncontrolled stress on encapsulated agent, which could be a real drawback. So to overcome all these limitations, droplet-based microfluidics appears as a good method and as a good tool. So what is droplet-based microfluidics? So this is a method, a tool which consists of handling two immiscible solutions again, but now at micrometric scale to generate microdroplet and to have more control. So basically, we will regulate precisely the flow rate of the two immiscible solutions, for example, oil and water here, and we will generate flow rate inside a micro environment, which is controlled, called microfluidic chip with a certain design, such as we can see on the pictures corresponding to a microfluidic chip with a certain nozzle. And the idea is that the dispersed phase will go to the central channel, the continuous phase on the other channel, and by regulating precisely the two flow rates of the two solutions, we will generate a squeeze in two dimensions to generate highly monodispersed droplets. So this technology appears with many advantages, such as better control over the process. As we are at micrometric scale, we have more control. We then increase the droplet monodispersity, and we have a better reproducibility. The microfluidic allows to make automation possible, so we can make a production continuously without interruption. By increasing the monodispersity, we avoid the loss of expensive reagents. And it is really easy to control the flow rate. So to have a flexibility on particle size, by changing the flow rate, it's really easy to make different batches of different particle size. The limitation of this technology is that this technology remains less known in industrial pharmacy, in industrial area. It is adapted to lab scale production for one single device. We are able to produce up to gram per day. However, to large scale production, this is another question, and this is one of the big challenges of the microfluidic today. So if we compare in terms of monodispersity of control, traditional batch appears with a dispersion of around 20% in terms of particle size, and microfluidic-based method allows to go down to 5% in dispersion, so really high monodispersity, which is one of the best advantages. But what this monodispersity means in terms of application. So if we focus on this article, where two particle sizes were generated, 11 micrometer and 41 micrometer, with two different techniques, the conventional method, which is the batch method that I presented, and the flow focusing method, FF, which is the microfluidic method. If we are focusing on the 11 micrometer particle size, so the empty square match with the conventional method and the full square match with the flow focusing method, so microfluidic method. If we compare the two, we can see that monodispersity has a big impact. We have a slower drug release for the microfluidic method, microparticle generated with microfluidic method, much lower initial burst, something that we want to avoid. We also have a big encapsulation efficiency with microfluidics. And also, again, it's easy to play with the particle size, so we have a good flexibility in terms of particle size. So I presented you the advantages of microfluidic in general, but today I would like to present you the microparticle production station that we have developed in collaboration with Sequoia. And I would like to show you an example that we made using this microparticle station for PLGA microparticles for drug delivery. So we used, to generate PLGA microparticle, we used the microparticle production station, which gather all the instruments that you need to make a first experiment with microfluidic and generate easily microparticles, from microscope to fluid handling solution and device droplet generator. And in terms of reagents, what we have used is the disperse phase, the PLGA, polylactic-glycolide acid, dissolved in a solvent which is the ethyl acetate at different concentration. And in continuous phase, we use water with 2% polyvinyl alcohol, which is a surfactant. So what is PLGA? So PLGA, first, is biodegradable and biocompatible polymer. It is widely used in drug delivery application to encapsulate agent pharmaceutical ingredients inside microparticle. This allows to have a drug delivery system which is non-invasive. We can use this microparticle via injection. And it is easy to functionalize these microparticles, this polymer, to allows to target specific sites for drug delivery application. So we are using, we used the ethyl acetate as a solvent. While all the articles are highlighting the use of dichloromethyl, we have decided to use ethyl acetate. Why? Because dichloromethyl is widespread in the microparticle production area. However, we have seen that it is really hazardous for API. As comparing to the ethyl acetate is a class 2 solvent, which is considered as less hazardous and should be used in pharmaceutical area. If we go in detail, so ethyl acetate is more biocompatible than dichloromethyl. We have seen that, according to articles, the API loss during precipitation time process is much lower than using the ethyl acetate than using dichloromethyl, and the precipitation time is in seconds for ethyl acetate, whereas it is for minutes for dichloromethyl. So that was for the materials. Now let's see the method to produce these microparticles. So the method could be split in two parts. First of all, the droplet formation, and then solvent evaporation. So to generate the droplet, we use the red drop droplet generator. So basically, this red drop is based on two glass capillary, axisymmetric glass capillary aligned together in a chamber which will be filled with continuous phase. If we focus on the nozzle on the right side, we have pictures with the nozzle and a droplet generation. So the dispersed phase, we will control the flow rate of the dispersed phase, which will go through the nozzle. We will also control the flow rate of the continuous phase. The continuous phase will then generate squeezing shear stress in all dimensions, which will lead to a droplet generation with a high monodispersity. Here, this technology, this is the geometry which is leading the droplet formation. So it allows to generate water in oil droplets or oil in water droplet, regarding of the physical property of the materials. And so we don't need to make any coating on the capillary, glass capillary. So no need any surface treatment on the glass capillary, and no surfactant are needed to generate the droplet. Surfactants are needed to stabilize the emulsion after production, but in this case, we do not need any surfactant to generate the droplet. This technology allows also to generate, to handle partially miscible fluids, such as solvent in water, which is really important in this application. And also these solutions, compared to other microfluidic device and chips, has standard connection, which will help the user to launch a first experiment without any problem. So this is for the droplet formation process. Now let's go to the scheme with the solvent evaporation. So if we go back, here we have the dispersed phase, which will be the PLGA with the ethyl acetate mixed with the API, so the drugs. And as a continuous phase, we will have the water with polyvinyl alcohol, so the surfactants. We will generate these droplets as I presented. And if we focus on the droplet, actually the droplets are containing PLGA, solvents, and also the drugs. In contact with the water, we will have the solvent which will spread out of the droplet. We will have a solvent diffusion in water. This will lead to a decrease of the droplet size for the same amount of PLGA. That will be a droplet polymerization by precipitation. And this is how we go from a droplet to a microparticles. So the microparticles at the end will gather PLGA and the API, so the drug. So as you can see, the solvent has a tremendous importance here in polymerization process because it can modify the polymerization time. It is directly in contact with the agent pharmaceutical ingredient, so it has to be more biocompatible. That's why we use ethyl acetate. And it also affects the drug encapsulation efficiency. So in terms of results, with this station, we were able to produce different types of microparticles. We have tested PLGA with different concentration from 0.5% to 10%. We have modelized the encapsulation of API with a lysozyme at 3%. And we are able to generate bit size from 10 micrometer to 50 micrometer with this technology, with a dispersion lower than 2%, dispersion in terms of droplet size. So we had really high monodispersed droplets, microparticles. And we have an encapsulation efficiency higher than 95%. If we compare the two technology, the traditional batch method, comparing with the microparticle production station, so first of all, dispersity, we are at 2% versus 20, maybe more, up to 30% of coefficient variation for the batch method. We have more control of the droplet size, microparticle size. We have small process footprint as we are using microfluidic instrument. We have a smaller process footprint compared to the batch method. We have a method which is reproducible and compatible with wider range of solvents. If we compare with other microfluidic technologies, because there is other technologies, microfluidic technology that allows to make microparticles, we are a little bit better in terms of monodispersity from 2%, more robust and plug and play because we have standard connection. And the big advantage of this technology is that we can generate emulsion or microparticle without any surfactant and without coating needed. And we also are able to handle easily less toxic solvents, such as the ethyl acetate. So I presented you the example with PLG and microparticles. However, this system is really versatile. It can be used for many applications and to make any emulsion and produce many microparticles. So, such as alginate microparticles, agarose microparticles, PLA, and other polymer, for example. We have demonstrated with current standard solution, let's say, mineral oil in water, water in fluorinated oil, solvent, ethyl acetate in water, dichloromethane in water. And also it is possible to make bubbles, air in fluorinated oil, so we have tested, and we were able to generate stable bubbles. In terms of perspectives, so this system is able to be changed. The nozzle can be adapted to many applications. So we are working to develop new technology to make double emulsion for core shell particle production for drug delivery again application, nanoparticle synthesis for liposome synthesis, for example, in drug delivery. And we are working on a solution for parallelization and scaling up the system to increase the production. So in general, so microfluidic appears with many benefits, such as a better control on particle size, which leads to more control on drug release. We have a more homogeneous drug distribution into the microparticle using this technology, which allows, again, to have more control on the drug release. High monodispersity, so more reproducibility, and we have an encapsulation which is optimized. Today, one of the big challenges of the microfluidic is that we are lab scale, but we are working to go further to industrial scale. So if you are interested to work with us in this solution, we'll be really happy to collaborate together to go further together and increase the productivity of this technology. So that's all for this webinar. Thank you for your attention. Now we will have, let's say, 20-minute question session. I wanted to remind you that you can download also application notes about the PLGA microparticle station on this webinar. You also have datasheet for the red drop, so the device to generate microparticle and droplet, and also a datasheet for the whole package, so the whole station. So thank you, everyone. Feel free to ask questions. No question for the moment. Don't hesitate. Any question, feel free. So we have a question. What is the dimension of agarose and alginate microgel? So for the moment, we do not have a result with this alginate microgel. So this is the perspective. So in theory, the red drop is versatile enough to generate microparticle and microgel for alginate and agarose. And we are working on this application, but for the moment, we do not have really strong data to give you the answer for this. But we are working on this, and that will arrive soon. Another question. You mentioned a surfactant is necessary for stabilization. Yes. So the role of the surfactant is to stabilize an emulsion after production. So if you want to keep your emulsion for days, or it depends, hours, you need to add a surfactant in your solutions. However, the technology that I presented do not need any surfactants for droplet generation. This is the small difference. What range of flow rates did you use to generate the microbeads from 50 to 100 micrometer? And for which viscosity? Thanks. So maybe I will let Adrien Dewandre answer this question. I do not remember exactly the flow rate, but anyway, we have the data sheet of the PLGA microparticle station, and we have the application note which gather all the details about pressure, flow rate that you need to generate a specific size of microparticles. Adrien, do you have any idea?
The typical flow rate for the droplet generation in the PLGA is about 10 to 20 microliter per minute for the dispersed phase. And for the continuous phase, it's about 100 microliter per minute. And the viscosity of the droplet phase is 2 or 3 times the viscosity of the water.
Okay, thank you, Adrien. We have another question. So to use fluorinated solvents, do you require coating of the microchannels? So we do, with this technology, we do not need any coating on the microchannel. This is one of the big advantage of this technology. I would like to know, during the emulsions, do the API is also going out of the microparticles? This is yes. So this is a good question. When we are making polymerization by precipitation, so the solvent is going out of the droplets, and sometimes the API could follow the solvent and go out of the droplets or out of the microparticles. So this is something that could appear. That's why we are using ethyl acetate, because the polymerization time is really low. So in seconds, the API is less likely to going out of the droplets, the microparticles.
Can I speak?
Yes.
Yes. In the case of the test with the lysosine, we found a very small amount of lysosine in the continuous phase, meaning that the encapsulation efficiency is very high. So the efficiency is higher than 95%. So a very low quantity of API is lost in the continuous phase because the process is very fast.
How stable the encapsulated particles are if go under high shear environment? What do you mean by high shear environment? Could you maybe specify? Instead that okay. You want to answer, Adrien?
Yes, maybe.
Yeah.
But in the case of PLGA, we used centrifugation without a problem. But in general, that will depend on the polymer. But we don't make a very broad test on the mechanical resistance of the particles. It will depend on the polymer. So it's not possible to give a general answer on this question.
Are microgel cross-linked in the channels? Adrien, you have experience with that, I think, so maybe you could answer also?
Yes, in fact, the microparticles, the solvent evaporation is enough in the channels, so in the microfluidics device, to reach a state where there is no coalescence, there is enough solidification to avoid coalescence. So this polymerization, this precipitation, can continue in the tank you use to collect the droplets. So the goal in the microfluidic system is to avoid coalescence. So you have to reach enough precipitation to avoid any problem, any clogging, any coalescence in the system. And then in other system, you can continue to reach the total polymerization of your system. That's true for all the particle produced in microfluidics. You need to reach a sufficient state of polymerization to avoid clogging and coalescence.
Okay, we have another question. Yeah. How do you manage to generate PLGA particles with hydrophilic API? Indeed, to my understanding, PLGA is not soluble in water, and other macrophysics techniques are based on double immersion processes.
Yes, in this case, double emission is the best way to do that, because you can just make a shell around the API in a water solution. That's true.
That's why we are working on modification of the nozzle to make double immersion as well.
For the range of droplets in this system, the red drop in the actual system, the nozzle is 30 micron diameter, and the collect capillary is 150 microns. So the size achievable is for the droplets, it's from 50 microns to 120 microns. But it's possible to adapt the system to change the size of the nozzle and the output capillary just by changing two parts of the system, keeping the main box. So you can adapt the system to produce droplets from 20 to 200, 300 microns. Yes, just by changing two parts of the system. The dispersity is assessed by measuring the diameter. Yes, of course. Mm. And the smallest particle size, okay, in this system, so the system you saw in this presentation, with the size of the nozzle and the output capillary fixed, the smallest size is of the droplets, it's 50 microns, and for the particles, for the beads with the shrinking, it's 20 microns. But we can adapt the system to produce 10 microns droplets diameter. It's possible, yes. It needs adaptation of the system, but it's possible. We made it. Can small molecules like amino acid and ATP enter the microparticles? Yeah, if you can dissolve this particle in the polymer and solvent solution, the droplet solution, probably it's possible to get it in the microparticles. Yes, because the solidification, the gelification, the precipitation, sorry, is very fast. So you will lost a few quantity of particles of API. I'm not sure I answered the question, because maybe the question is if it's possible to get small molecules entering the microparticles when they're rigid. So you were speaking about the porosity or of the system. Okay, maybe we can answer this question later.
Yeah, it's something that if you have a specific question, we can just have a discussion later and we will see how we can do this together. This is possible. Okay, did we miss some questions? Because we do not see any more questions. So we will wait a few minutes. And if there is no more questions, we will close the session. So no, this is the advantage of this technology. So compared to other microfluidic chip glass in glass, here, if you have a problem, so if you clog the nozzle, there is many ways to clean it. So we have everything detailed in the application notes. So if you want to watch it, so this is first you can try to clean it with many methods. We can put it in ultrasound baths if it's possible, and if it's the nozzle is broken, we can just change the nozzle and you will replace it and you will have a fresh new nozzle actually. So you do not need to change the whole setup if you want to, if you break the nozzle.
Okay, so there seems to be no other questions, or anyway, if you have any that comes up, please feel free to send us your question at contact@fluigent.com, and we'll be happy to answer you then. We want to thank you again for your participation. We hope you enjoyed it. We remind you that the recording of the presentation will be available from tomorrow. You should receive an email from WebKO platform with a link to the recording. Please also have a look at our dedicated page for the webinars, for the list of upcoming webinars. And with that, also don't forget to download the documents that we have available for you to on the webinar. We wish you a pleasant day and see you soon. Bye.
Bye.